In this video, we are going to conclude the topics in chapter 13. Now we're going to talk about artificial sweeteners, and we are also going to be introducing and talking about polysaccharides. So, some people are particular fans of artificial sweeteners. And artificial sweeteners, when they were created, it was providing a way in which you can still have a sense of eating something sweet, but you were not going to be converting these sugars into anything that produce energy. That's why they're called no calorie sweeteners. And I'm going to talk about a few of the ones that are the most popular out there. So when it comes to artificial sweeteners, the first one that I want to talk about is sucralose. And sucralose, which is marketed as Splenda, at first when it was inspired on its structure, it was derived from the molecule of table sugar. So table sugar is the molecule that we have on the left. And as you can see, the difference between table sugar and sucralose is actually 1, 2, 3 chlorine atoms that are present in the molecule itself. So they just replace those hydroxyl groups by chlorine atoms. In a later table, we're going to see how the change of those chlorine atoms are going to be changing the levels of sweetness for this particular molecule. Understand that artificial sweeteners are not bad to consume, but you have to do it in moderation, because it has been shown time and time again that, at least no studies in human, but in animal models, that these artificial sweeteners are actually creating addiction, and in some cases liver damage, kidney damage, and even creates, if consumed in really high amounts, seizures. Let's move on to the next artificial sweetener, which is aspartame. So aspartame is what we consider in the market as NutraSweet or equal. Now, this one is considered a non-carbohydrate sweetener because when we look at its structure, it doesn't even look like a monosaccharide. One of the things that is a special feature in aspartame is going to be that it is actually made by linking 2 amino acids. And we're going to talk about amino acids later in the semester. The 2 amino acids that are present in aspartame is aspartic acid and phenylalanine. And in phenylalanine, at the end of the molecule, they incorporated a methyl ester. Now, this is one of the main sweeteners that is used out there. So if you drink Diet Coke or many diet products, that's the one that it is present. So if you see that in the food label it says contains phenylalanine, most likely aspartame is one of the sweeteners that are present. Now, one of the things is that when it comes to sweetness levels, understand that these artificial sweeteners are going to be, when compared to table sugar, very sweet. So they were intended to be consumed in very small portions, but right now people actually use them in excessive amounts. Let me show you in table 13.2 in your textbook. If we compare different monosaccharides or disaccharides or even sugar alcohols to table sugar, how does that affect the sweetness level? So let's say that we consider that the sweetness level, and I'm just going to highlight it in red, for sucrose, which is table sugar, is 100. The only monosaccharide or disaccharide that it is sweeter than table sugar is going to be fructose or fruit sugar. Now, when it comes to the sugar alcohols, notice that xylitol, which comes from the monosaccharide xylose, maltitol, just to review, comes from maltose. So xylitol is the only one that is as sweet as sucrose. Any of the artificial sweeteners, aspartame, saccharin, sucralose, or neotame, as you can see, if we compare it in the scale, it is very sweet compared to table sugar. So again, these artificial sweeteners were intended to be eaten in small portions, but it's just that people keep on consuming them in high amounts, and to date we really don't have the studies to back up and understand what are the effects in the body. And because sometimes people are skeptical of the use of animal models, they're like, oh, that was done in a different system that is not human, so how is it going to relate to me? Understand that animal models is the beginning of research, and it is very relevant. To give you guys an example, a rodent and a human share about 90% of the same genes. That's why when research is done in small animals, rodents is one of the ones that is utilized, and at times it can be, okay, or it may be related to the effects that happen in our body. Another important link between carbohydrates in our body is going to be the different blood types. I do not require you guys to know what are going to be the monosaccharides that determine the O type, the A type, the B type, and even the AB type of blood. But I just want you to know that the way that blood types are determined is by looking at the terminal carbohydrates that are in the surface of red blood cells. The last topic in chapter 13 is going to be polysaccharides, and understand that polysaccharides are going to be many units of monosaccharides that are going to be linked with one another. Now, all the polysaccharides that we are going to be talking about in chapter 13 are going to be polymers of glucose. So let's talk about the first polysaccharide, which is starch. Okay. Starch is composed of two different polysaccharides. The first one is amylose and the other one is amylopectin. So understand that the difference between amylose and amylopectin is that amylose is a straight chain polysaccharide of glucose units, while amylopectin is going to be a branch chain of glucose. So as you can see in the image towards the left, this will be the structure of amylose, while the bottom one is going to be the image of amylopectin. Where do we find starch? In any starchy food. And it is necessary in our diets because this is a source of glucose, because we are able to hydrolyze these starches into glucose and we can derive energy from these products. Now, glycogen, understand that it is a polymer of glucose, and it is important for us because this is how we store in liver and muscle glucose. We don't store a lot of things, but as we're going to see, we store fat and we store sugar. We do not store amino acids, but we're going to learn that later in the semester. So glycogen, when it is hydrolyzed, is what provides and maintains blood glucose levels. And because glucose is utilized to provide energy by the reaction that was discussed in the beginning of today's chapter, that's why it is important that we have it. In terms of the structure, understand that glycogen is very similar to amylopectin because it is branched, and it is actually highly branched. The last polysaccharide that we're going to talk about is cellulose. Understand that cellulose is what gives the rigid structure to cell walls in, for example, products like wood and fiber. Now, this polysaccharide, even though it contains glucose, okay, is unbranched. Now, this polysaccharide cannot form hydrogen bonds in water, so that means that it is insoluble in water. Understand that cellulose, if we put it in our body, so for example, if you eat anything that contains fiber, we do not have the capacity to hydrolyze cellulose. So when we eat fiber, what it does is actually pushing things outside the body. So understand that cellulose does not contribute energy to humans, and it is precisely because we cannot hydrolyze it, okay? We cannot digest it. So I know that out there, people sometimes say, oh, I'm doing the celery diet because celery has negative calories. People, that is a myth. The only thing that celery is giving you is water, because the fiber that you're obtaining by eating celery, all it's doing is pushing things out of the body much better, because we are not spending any energy to convert that fiber into glucose, because we do not have the biomolecules that can do that reaction.